Nucleus, house of coded instructions. The nucleus has double-layered covering called nuclear membrane. Nuclear membrane has pores that allow the transfer of material between nucleus and cytoplasm. Nucleolus is present in nucleus as dense round body, and in these nucleolus, synthesis of ribosomal subunits take place, and these ribosomal subunits exist from nucleus to cytoplasm. In cytoplasm, one large and one small subunit assemble to form ribosomes. In nucleus, when cells are about to divide, chromosomes become visible as thread-like structures. Chromosomes contain DNA, deoxyribonucleic acid, molecules which contain the genetic information. The functional segments of DNA are called genes. In a non-dividing cell, DNA is present as a part of chromatin material. This chromatin material is visible as entangled mass of thread-like structure. This is a structure of nucleus, nuclear pore, nucleolus, nuclear membrane, chromatin, and nucleoplasm. This is a structure of nucleus containing chromatic materials and the chromosomes. Enucleate means without nucleus. Mature red blood cells in human body do not have a nucleus. Why they do not have a nucleus? Because they need the space for the hemoglobin, and hemoglobin transport oxygen to all the cells of the body. So due to make the space for the hemoglobin, they are enucleate, यानी कि they are without nucleus. The absence of a nucleus provides more space for hemoglobin, allowing it to transport larger amount of oxygen to all cells of the body. They cannot repair they themselves, nor they can divide as they lack nucleus. They have lifespan of 120 days. And this is the structure of red blood cells. We have bioconcave discs, hemoglobin, and cell membrane. We are not seeing the any nucleus in it. Now, what is a nucleoid? Nucleoid Nucleus in prokaryotic cell is called nucleate. These cells do not have well-defined nucleus. Their DNA is present as a single circular molecule. These circular molecules are associated with specific proteins. The regions containing these genetic material is called nucleide. Smooth endoplasmic reticulum, SER. It does not have a ribosomes on its surface, therefore looks smooth. It is involved in the synthesis and storage of fats and hormones. Golgi apparatus, the packaging and shipping center. These are stacks of flattened sac-like structures. Together, these stacks are called Golgi apparatus, as you are seen in the figure. It is functionally linked to ER, that is endoplasmic reticulum, cell membrane, and other cell organelles. It modifies, sorts, and packages proteins and lipids into vesicles from transport secretions and lysosome formation. Now, what are the lysosomes? These are cleaned up system of the cell. These are single membrane bound sacs filled with enzymes. They can break down unwanted materials like carbohydrates, fats, and damaged parts of a cell. The product formed by the breakdown are released into the cytoplasm where they may be used in other cellular processes. Lysosome enzymes in human sperm cells helps to break down the outer layer of the egg, allowing fertilization. Mitochondria, the powerhouse of the cell. It supplies energy needed for cellular activity. That is why it is called the powerhouse of the cell. Each mitochondria has two membranes. The outer membrane is smooth and porous. The inner membrane is folded into finger-like projections called cristae. The increased surface area for chemical reaction and facilitate energy production. During cellular respiration, glucose and other molecules break down to release energy. This energy is stored in the form of molecule called adenosine triphosphate (ATP). ATP acts like energy currency and used for most cellular activity. Plastids, center of food synthesis in plant cells and beyond. Plants use special organelles called plastids for food synthesis and storage. Now, the most common plastid is chloroplast in the plant cell. Chloroplasts are the double membrane bound organelles like mitochondria. Chloroplast has semi-fluid substance called stroma. Inside stroma are disc-shaped membrane structures that contain chlorophyll, and this chlorophyll absorbs light during the photosynthesis. Sugar synthesis is stored in stroma along with starch granules. Mitochondria and plastids have their own DNA and ribosomes, thus they can make some of their own proteins. This suggests that both mitochondria and plastids share evolutionary history with single-cell organisms like bacteria. As you are seeing in the figure, we have two kinds of membrane, outer membrane, inner membrane, stromal lamella, stroma, lipid droplets, ribosomes, grana, and starch granule. This is the electron micrograph section of chloroplast. Vacuoles, the organelles for storage and support. These vacuoles are commonly present in the plant cell in very large size. But in animal cell, we do not need the storage of food, so they are very small in size. In plant cell, there is one large central vacuole surrounded by a single selectively membrane. Vacuole stores water, minerals, and sugars and waste materials. It stores large amount of water to maintain pressure inside the cell. When plant does not get water, it gets wilted as vacuole loses water. In animal cells, small sized vacuoles are present and they help in temporary storage of material.